A mammal can feed its growing embryo continuously through a placenta. A plant, however, has to pack its offspring a self-contained lunchbox, the seed, before sending it out into the world. Angiosperms, or flowering plants, solve this with a highly efficient mechanism called double fertilization. It perfectly synchronizes the creation of the baby plant with the creation of its food supply. Looking at this pistil cross-section, reproduction starts on the stigma, travels down the style, and ends inside the ovary. Here sits the embryo sac, holding two distinct fertilization targets, an egg cell near the bottom and a pair of polar nuclei in the middle. Knowing exactly where these specific cells sit is the first step to understanding how the plant executes its 2-for-1 reproductive strategy. This diagram shows a pollen tube actively growing. After a pollen grain lands on the stigma, it bores down through the style, acting as a protected highway to the ovary. It is easy to assume one pollen grain carries one sperm cell, but watch the tip of the tube. The pollen's generative nucleus has actually divided to form two distinct male gametes traveling side by side. When the tube reaches the ovule, it enters through an opening called the micropyle. It pushes into one of the synergid cells, ruptures, and releases both of those male gametes directly into the cytoplasm. Unloading two separate gametes into the exact same microscopic staging area sets off a synchronized dual-target chemical reaction. Let's track the first of those two gametes. It leaves the synergid, moves over to the egg cell, and the two nuclei fuse together. This primary fusion is called syngamy. One haploid male gamete joins one haploid egg to form a standard diploid zygote. But we still have that second male gamete. It bypasses the egg entirely and migrates to the large central cell to meet the two polar nuclei. Take a second here. If one haploid gamete is about to merge with two separate haploid polar nuclei, what is the final math? 1 + 1 + 1 = 3. They form a triploid structure, known as the primary endosperm nucleus. This specific event, triple fusion, is how the plant builds a dedicated food storage system that is biologically distinct from the embryo itself. Looking at these study notes on post-fertilization events, we can see exactly what happens to those microscopic fusions as the ovule matures into a seed. First, the diploid zygote divides and differentiates. This becomes the literal plant embryo, the future root and shoot. Meanwhile, that triploid primary endosperm nucleus undergoes repeated divisions to form the endosperm. This acts as the nutrient-dense reserve, effectively feeding the embryo while it develops. We can summarize the entire mechanism with this graphic. Syngamy creates the diploid zygote, and triple fusion creates the triploid endosperm. Together, they make up double fertilization. By inextricably linking these two events, the angiosperm guarantees it never wastes resources building an embryo unless it has also packed the exact fuel for it to survive.